Welcome back to this next video in which we are discussing control of blood pressure and blood volume. Okay, so we've now finished discussing the baroreceptor response, which was this very quick feedback mechanism for returning mean arterial pressure to what it was after you've had some deviation from normal. So either it's gone too high or it's gone too low. We're now going to discuss another feedback mechanism for returning mean arterial pressure to what it was, which is the Renin-Angiotensin-Aldosterone System, usually abbreviated just down to the RAAS for short. Okay, now this is going to be a slower response than the baroreceptor response, and it's going to return blood pressure to normal in a slightly different way to the way that the baroreceptor response did. So let me just go over this. So let's start off with the case that mean arterial pressure has gone down. So mean arterial pressure is below what it would be normally. In this case, you're going to get activation of the Renin-Angiotensin-Aldosterone system. So the system becomes activated when mean arterial pressure goes down. It's going to become inhibited beyond its basal level of activity if mean arterial pressure goes up. So the two are obviously going to have utterly opposite effects. So what then, once the Renin-Angiotensin-Aldosterone system has been activated, is actually going to happen? How is it going to return mean arterial pressure back to normal, i.e. raise it back up? Well, it's going to do two things. In the short term, two major things. It does a lot of things, but the two major things is, in the short term, it is going to constrict blood vessels. And you might think, haha, it's going to constrict the arterioles to the skin, the gastrointestinal tract, and the skeletal muscle. All of what we've seen before. It will raise total peripheral resistance. I completely understand that. But actually, it's going to do something more generalized. It's going to produce vasoconstriction all over the body. And in fact, the Renin-Angiotensin-Aldosterone system is going to be capable of vasoconstricting all of the great veins of the venous system. Now let's just think about this. If I bring back up my basic model of the cardiovascular system here, effectively we're going to constrict all of the great big blood vessels all over the body, particularly in the venous system. So if you can imagine constricting all of the great veins in the venous system, and of course it will constrict the arterioles as well, which is going to be important for raising total peripheral resistance. But if you think about constricting the great veins of the venous system, what that's going to do is it's going to raise mean venous pressure. And we know that raising mean venous pressure is going to raise end diastolic volume in the heart, which will then raise cardiac output by raising stroke volume. So actually, by constricting the venous system down, we are going to effectively raise the mean systemic pressure, and that's going to help us bring cardiac output back up. So we're not just going to constrict arterioles to raise total peripheral resistance for blood moving from the arterial system to the venous system to raise arterial pressure. We're also going to constrict all over the vasculature. And you know, basic common sense, if you constrict the entire vasculature, of course blood pressure is going to go up. Of course, by blood pressure I mean the pressure in the arterial system. So that's it's more generalized effect than just constricting arterioles. So this vasoconstriction effect then, it's going to have two major things that it's going to produce then. It's going to raise total peripheral resistance, and we understand that that's exactly what the increase in sympathetic tone to the arterioles was doing in the case of the baroreceptor response. But it's also going to result in vasoconstriction of the venous system, and that's going to result in a rise in mean venous pressure, and we know that an increase in mean venous pressure is then going to lead to cardiac output going up. Okay, so that's one of the major things that the Renin-Angiotensin-Aldosterone system is going to do. It's effectively making it so that the volume of blood that you've got in the vasculature is going to be more sufficient for keeping a high blood pressure. So that's one of the major things that the Renin-Angiotensin-Aldosterone system is going to do. In addition to that, it's also going to actually work to raise blood volume. So it's going to increase blood volume, and of course if we increase blood volume, we know exactly what will happen there. That will cause the amount of blood effectively in the venous system to hugely increase, and therefore mean venous pressure will increase, end diastolic volume will increase, stroke volume will increase, cardiac output will increase, and therefore mean arterial pressure will increase. So these are the two things that I want to understand about the renin-angiotensin-aldosterone system. How is it going to cause vasoconstriction all over the body in arterioles, in addition in arteries as well, which will directly raise mean arterial pressure, but also in the venous system to increase mean venous pressure and thereby increase cardiac output, and also how is it going to raise blood volume. Right, there are other effects of the renin-angiotensin-aldosterone system, but these are the two major ones which we'll focus on. That's what happens if mean arterial pressure goes down. If mean arterial pressure goes above what it should be, then what you're going to get is an inhibition of the renin-angiotensin-aldosterone system. So the activity of this system will fall, and that will then mean that blood volume will go down and you'll get vasodilatation. So you'll get the opposite thing. So there's usually a basal amount of activity in this system. It's usually on at a very low level. We're going to lose that renin-angiotensin-aldosterone system tone, if you like, and that's then going to cause the opposite effects to what it leads to when it's activated. So we'll get a little bit of vasodilatation, which will lower total peripheral resistance, lower mean venous pressure, and thereby lower cardiac output. And we'll also get a reduction in blood volume, so we'll lose more fluid from the body. Okay, so the exact opposite things are therefore going to happen when mean arterial pressure is too high. Okay, so what I now want to talk about then is, what does the renin-angiotensin-aldosterone system actually involve? What are the components of it? So it's one of these absolutely beautiful endocrine pathways where one thing activates another thing, and it forms a beautiful flow diagram. So the name of the system gives it away here. We're going to start off with renin. Renin's the first thing that's going to be activated in the renin-angiotensin-aldosterone system. Then it will go on to angiotensin and then aldosterone. So we're going to work our way through these. And really, it's best to take the case of mean arterial pressure going down, because that's what's going to activate the renin-angiotensin-aldosterone system. So we'll look at this top one, and then we'll talk about the bottom one in the end, and we'll talk about how the opposite things will happen, how the renin-angiotensin-aldosterone system, its tone, its basal level of activity is going to be toned down when the mean arterial pressure goes up, and that will lead to the opposite effect. So let's start by taking the top one here, that mean arterial pressure has gone down. Let's say you've become dehydrated, or you're suffering from septic shock or something along those lines, or hemorrhage. So mean arterial pressure has fallen, and we want to see the activation of the renin-angiotensin-aldosterone system. So this system is completely centered around the kidneys. So the first thing that I want to do is just discuss some basic renal physiology with you, because the kidneys are often something that people's knowledge isn't particularly good about. So what I want to do is draw firstly a large picture of a kidney and look at the different portions of the kidney. And then I want to show you the functional unit of a kidney, which is a nephron. And then once we've gone over the basic principle of what the kidneys are actually doing, and don't worry, we won't be doing it in full detail because you can, have weeks and weeks of lectures on the kidney, but we'll just have the basic overview of what the kidneys actually do. Then what we'll do is we'll specifically study a structure within the functional unit of a kidney, which is obviously the nephron, called the Juxtaglomerular apparatus, and that's the apparatus that is going to detect a fall in mean arterial pressure, or indeed a rise in mean arterial pressure, and is going to adjust its release of the enzyme renin, the start of the renin-angiotensin-aldosterone system, accordingly. Okay, so starting off then, basic renal physiology. So what I'm going to do is I'm going to draw a kidney. We'll go for the right kidney, and I'm going to have taken a coronal section through the right kidney. So if you imagine the position of the kidney against the posterior wall of the abdomen, then we're going to take a coronal section, a nice slice through the kidney, and we'll be able to see the inside portions. Okay, so I'll draw it like so. Okay, in this classic shape here. Okay, so here is my right kidney, and let's also have the ureter, the right ureter, coming off here. So let's just label up portions. So this is the ureter. Okay, right, the right ureter in particular. I'll just label it up, right ureter. Okay, so obviously the right ureter carries the urine that the kidney has produced from the kidney to the bladder. Okay, so starting from the outside then, the kidney is covered by a capsule. So I'll start by putting on the capsule. I think I'll colour in the capsule in pink here. So around the entire outside then of the kidney, you're going to have a capsule which is known as the renal capsule. So that's the starting point. We're going to start from the outside and work inwards. I think that's the best way of doing this. So this in pink, this is the renal capsule. Okay, now going inwards, so inside of the renal capsule, what do you have inside the renal capsule? And this is where the important tissue actually begins. The renal capsule is just there for protection. Okay, the actual functional tissue is now within the renal capsule. Okay, so the actual functional tissue then of the kidney, which is going to be filtering the blood. So the basic function of the kidney is to filter the blood, remove waste products from the blood, such as urea, for instance, is the classical example. So toxins, poisonous molecules that the body is producing all the time, which we want to have removed from the blood continuously. It's going to be filtering these out and putting them into the urine. So the functional portions of the kidney, the actual tissues that are performing this function, are split into the cortex tissue and the medulla tissue. So cortex is a Latin word, I believe. It means the outer portion of something. So for instance, the cortex of an orange would be the orange peel. Medulla is another Latin word meaning the middle of something. So the medulla of an orange would be the nice juicy bit inside. So we'll start by drawing the renal cortex. So this is the portion that's just underneath the renal capsule. Now the renal cortex, it has extensions that go into the renal medulla portion, which have the same function as it. So I'll draw some of these at the same time. So here we go. So here's the renal cortex, and here's one of these extensions inwards like so. And these extensions inwards to the medulla are known as renal columns, but they're functionally equivalent to what the cortex tissue is doing. Okay, so like so. So there's another renal column. Okay, so this portion then is renal cortex. So all of this is the renal cortex. Now drop the word renal and just put cortex. Just straighten it up a bit because it's gone on a funny angle. Okay, so cortex. So we'll have the cortex coloured in green. So all of this portion is the renal cortex, and these extensions that we've got inwards, those are called renal columns. Now they are considered part of the medulla simply because of their positions, but effectively they're just continuations of the cortex. They contain the same sort of stuff as the rest of the cortex here, so in green there. So these extensions inwards, these are the renal columns. And I will explain what actually the renal cortex consists of, what sort of tissue is going to be in the renal cortex and also in the renal columns. Once we've looked at the nephron, I'll show you which portions of the nephron are going to be in the renal cortex and the renal columns. So this is a column here. Now, in between the renal columns then, you have the other more characteristic portion of the medulla, which is the renal pyramids. So I'll draw a few of these on now. So here are some renal pyramids. So these are surrounded by renal columns, and they are called renal pyramids because they have a sort of pyramidal shape, like so. And these are the sort of prototypical portion of the renal medulla. So this is a renal pyramid here. Okay, right. And I will colour in the renal pyramids in red, like so. So these are all renal pyramids. Then the centre portion is where the urine that the tissue of the kidney is actually producing is going to collect and then drain into the ureter here. And this is known as the renal pelvis. So this is the pelvis of the kidney, like so. And I'll underline the renal pelvis in an orange. And this space here is continuous with the ureter here. Okay, right. So now then, let's actually see the functional unit of the kidney. So this is what you would see if you were to chop through a kidney and have a look at the new exposed surface that you've produced. What we are actually interested in is the functional unit of a kidney and what this tissue is actually made up of in terms of the functional unit of the kidney. So the functional unit of the kidney is known as the nephron, and you're going to have a huge number of nephrons in a single kidney. I think it amounts into the millions. So you have a huge number of these functional units known as the nephrons. And the job of a nephron is to filter the blood and produce urine. And the basic principle of how a nephron works is that it is going to produce a filtrate from the blood. So it's going to have a bunch of capillaries at the start of it, and I'll show you these capillaries in a moment. And what's happening is that you're taking a lot of the fluid out of the blood that is passing through these capillaries, and it's going to go into the nephron. Now, initially, this filtrate that is produced, known as the glomerular filtrate, this contains loads of stuff that we have absolutely no intention of throwing into the urine. So the principle then of the rest of the nephron, the function of the rest of the nephron, is to reabsorb loads of the stuff from this glomerular filtrate that we want to keep in the body, and also to secrete in lots of things from the bloodstream that we really want to get rid of. So let me now show you the different portions of a nephron. So the nephron begins with what's known as Bowman's capsule, and I will go into the structure of Bowman's capsule in a lot more detail in a moment on another picture, because we're going to need to study Bowman's capsule in order to see where the juxtaglomerular apparatus, which is so central to the Renin-Angiotensin-Aldosterone System, is. But for now, this is a basic picture. So here is Bowman's capsule. And you're going to have in this space here of Bowman's capsule lots of capillaries. So you'll have an afferent arteriole coming in here, splitting up into loads of capillaries, like so. And all of these capillaries are going to be producing a filtrate. So fluid is going to be filtering from the kidney, from these capillaries into this space in the Bowman's capsule, and this is going to be the glomerular filtrate. So let me just colour this in a little and add a few labels on here. So I'll colour this all in red because they're all blood vessels here. Okay, and unfortunately I haven't done it. I didn't do it finely enough to get those sort of gaps in the middle not coloured in, so now it just looks like one great big blob, but those portions in the middle were clearly not supposed to be coloured in, so I was supposed to have 3 capillaries shown here on this picture, but never mind, we didn't quite get that. Okay, so this is the afferent blood vessel coming into, splitting into the capillaries here, and this is known as the afferent arteriole, okay, like so. And the other blood vessel coming out is known as the efferent arteriole. It's not called a venule because it's actually going to split, it's going to drain other tissues still, so it's not the end of this blood vessel. It's now not just going to, find other blood vessels to merge together with and drain into the venous system. It's got other interesting things to do, so we don't call it a venule. Instead, it's known as the efferent arteriole. Okay, so it's the arteriole leaving the glomerulus, which is the bunch of capillaries in between the afferent arteriole and the efferent arteriole, but it's certainly not a venule. Its blood has not been deoxygenated yet, and its blood is not just going to go back into the venous system at all. It's going to go elsewhere. Okay, so afferent arteriole, efferent arteriole, and this cluster then of capillaries that the afferent arteriole has split into, and which are then reconverging up to form the efferent arteriole, this is then known as the glomerulus. Okay, so the glomerulus means all of these capillaries, and if you wanted to refer to a single one, you'd call it a glomerular capillary. Okay, so the basic principle then is that the afferent arteriole brings blood in, the blood is going to go through the capillaries of the glomerulus here, and as it does so, fluid is going to move from these glomerular capillaries into the space of Bowman's capsule, which is known as Bowman's space. Okay, so this here is the Bowman's space inside the capsule. So Bowman's capsule is just lined with cells, and then inside you've got a space known as Bowman's space, and we'll be studying the cells that line Bowman's capsule in more detail later on. So I won't go into any more detail here. We'll do that in a more in detail picture. So Fluid then is entering Bowman's space, and that fluid is now called the glomerular filtrate, and that fluid is going to be very similar to the plasma of the blood. It's not going to be identical to the plasma of the blood. Some proteins that are in the blood plasma are not going to go into Bowman's space. In particular, negatively charged proteins such as albumin do not end up in the glomerular filtrate, but a lot of other things, proteins don't generally end up in Bowman's space, but a lot of other things do end up in here. So for instance, all of the electrolytes are going to end up in the glomerular filtrate, along with, of course, the water that makes up the fluid. So a lot of good stuff. In addition, glucose can end up in the glomerular filtrate. So a lot of good stuff that we do not want to get rid of, and small little proteins can end up in the glomerular filtrate, and amino acids certainly can end up in the glomerular filtrate. So lots of good components of the blood that we don't want to get rid of do end up in the glomerular filtrate. Okay, so we produce this glomerular filtrate from the glomerulus then, and now the principle is that this glomerular filtrate is going to make its way through the nephron, and we're going to, along the length of the nephron, absorb lots of the good stuff back, and then throw back in, secrete out from the cells of the nephron a lot of stuff that we want to end up in the urine. So now let me show you the different portions of the nephron. So we begin with what's known as the proximal convoluted tubule, hence why I'm drawing it as a squiggle like so. So this is what's known as the proximal convoluted tubule, often abbreviated down to PCT for short. So the P is for proximal because it's close to the Bowman's capsule. The C is for convoluted, and the T is for tubule. Okay, and by the way, all of this is going to be within the cortex of the kidney, or in one of the renal columns. So in the cortex tissue or the renal columns, all of this tissue shown in green here, you're going to have glomeruli with Bowman's capsules and then proximal convoluted tubules coming off them. So if I wanted to add this onto this picture, I could have one here, for instance. Here's the Bowman's capsule, here's the proximal convoluted tubule, and I'll draw the other bits on when we get there. Then what's going to happen after the proximal convoluted tubule, and I'll just colour code this, we'll have the proximal convoluted tubule coloured in yellow here. Okay, after the proximal convoluted tubule, where loads of things are going to be reabsorbed from the glomerular filtrate back into the bloodstream, and lots of things are also going to be secreted from the bloodstream into the glomerular filtrate as it passes through the proximal convoluted tubule, so the fluid's going to pass in this direction. After the proximal convoluted tubule, we're then going to have what's called the loop of Henle, where the tube of the nephron loops down into a renal pyramid, into the renal medullary portion, like so. Okay, so it's going to loop down like so, and then come out as well. So you have this nice loop here, which is known as the loop of Henle, LOH for short. So this is the loop of Henle, named after the first person to discover it. Right, so the loop of Henle then is going to be this loop down into one of the renal pyramids. So if I draw that then on this picture, we're going to have something like this. Here is this loop down into the renal pyramid, and then it comes back up out into the cortex or column tissue again. Okay, so you can split the loop of Henle then into the descending loop of Henle, which is known as the DLH for short. You could put DLOH if you wanted, but DLH is the more usual abbreviation for the descending loop of Henle, so you get rid of the of. D is for descending, LH is then for loop of Henle. Okay, so all of this portion where we're going down into the depths of the renal pyramid, that will be the descending loop of Henle. Then the ascending loop of Henle is this portion here, where we're ascending back out of the renal pyramid up to the cortex again, and the ascending loop of Henle is split into two bits. So down the middle here, the first portion here is known as the thin ascending loop of Henle. So you put a lowercase t here, and then you have ALH again for ascending loop of Henle. So thin ascending loop of Henle. So the little t here stands for thin ascending loop of Henle. And I'll just colour in the thin ascending loop of Henle here in orange. Then, after the thin ascending loop of Henle, this other portion of the ascending loop of Henle, this is known as the thick ascending limb of the loop of Henle. Oh, by the way, you can take, you can give them even more pretentious titles, so you could call this the descending loop of Henle, or you could call it the descending limb of the loop of Henle, so people like to stick the word limb in, which is what we do here. So, particularly with this portion of the ascending loop of Henle, we call it the thick ascending limb of the loop of Henle, so T-A-L, really, this stands for thick ascending, that's the A, and then L is for limb rather than loop of Henle, and then it would be of the loop of Henle. So we divide the ascending portion of the loop of Henle then into the thin ascending limb of the loop of Henle and the thick ascending limb of the loop of Henle. Okay, then after the loop of Henle, then you're going to have another convoluted tubule, which actually ends up being very close to the initial glomerulus and Bowman's capsule, as we'll see later, but for this picture I'll have it drawn over here, so in reality we'd like this to be close here, however. So what we'll have then is another convoluted tubule known as the distal convoluted tubule this time, because it's far away as far as the nephron is concerned from the Bowman's capsule than the glomerulus. However, actually this ends up being very close to this. You can imagine that in three-dimensional space, okay, in a two-dimensional picture I've drawn them far apart, but in three-dimensional space this doesn't need to be so far away from this. So this is the distal convoluted tubule, the DCT, and then what's going to happen is the distal convoluted tubule is going to then drain into a collecting duct, like so, at which lots of other nephrons will be draining in, so there'll be lots of other distal convoluted tubules. So here might be another distal convoluted tubule of another nephron draining into this collecting duct here, and the collecting duct will then just go down and drain into the renal pelvis here. So drawing this on, the collecting duct might just come like so. Okay, and the collecting duct can be split into two portions. There's the portion of the collecting duct that is still in the renal cortex, and it's best to look at this picture for this. You can see this portion is in the green portion, it's still in the renal cortex. That's known as the cortical collecting duct, so the CCD for cortical collecting duct. And then there's the portion of the collecting duct that is in the medulla of the kidney, and that's known as the medullary collecting duct, and that would be called the MCD down here. Okay, right, so here are the different portions then of the nephron. So you're going to have absolutely loads of these nephrons in the kidney, and the principle, as I say, is that at the Bowman's capsule you are producing this glomerular filtrate, which is this ultrafiltrate of the blood plasma, which excludes, of course, the blood cells. The blood cells aren't going to end up in the glomerular filtrate. It excludes most proteins from it. All the tiny molecules are going to end up, so amino acids, glucose, the electrolytes, they're all going to end up in it. Some very small little peptides might also end up in it. So a lot of good stuff that we don't necessarily want to pass away in our urine is going to end up in the glomerular filtrate. This is then going to make its way through these nephrons, and in the length of the nephron, loads of the good stuff is going to be reabsorbed. So the amino acids, the glucose will be reabsorbed, most of the electrolytes will be reabsorbed, and then things that are bad are actually going to be actively secreted into the fluid that is moving through the nephron. And then what we end up with at the end of the medullary collecting duct, and it's important to say that although this is called the collecting duct, it does still have reabsorbative and secretory roles. The cells that line these ducts do still function to alter the composition of the stuff that's passing through them. Okay, the stuff that then finally passes into the renal pelvis, that's going to then be the urine. So that's the basic anatomy and physiology of the kidney then. Now what I want to talk about is the juxtaglomerular apparatus, which is the apparatus of the Renin-Angiotensin-Aldosterone System. It's the starting point for the Renin-Angiotensin-Aldosterone System. And the juxtaglomerular apparatus is found in all of the nephrons. So let me firstly just put the title, and then I'll actually draw you a nice picture of it out. So we're now going to talk about the juxtaglomerular apparatus, and as I say, this is a microanatomy structure, so it's a histological structure really, that you will find in all of the nephrons. So go to any nephron, you'll be able to find a juxtaglomerular apparatus. So when we talk about the juxtaglomerular apparatus, we're talking about one nephron, but of course in the context of the Renin-Angiotensin-Aldosterone System, you're going to have all of the different nephrons, juxtaglomerular apparatuses, becoming activated when mean arterial pressure goes too low. And they are going to then release the first enzyme, as it turns out to be, of the Renin-Angiotensin-Aldosterone System, which is of course renin. Okay, so the juxtaglomerular apparatus then. So this is actually going to involve a portion of the distal convoluted tubule, known as the macula densa, some of the cells that line the proximal portion of the distal convoluted tubule, this portion that I'm pointing to here, and also the glomerulus and the Bowman's capsule hugely. Particularly the cells that surround the afferent arteriole. So what I'm going to now do is I'm going to have a look at the glomerulus and Bowman's capsule. And by the way, there is a term that I'm dying to use, but which I haven't introduced you to, so I'm continuously having to tell myself not to use. So I'm going to introduce you to this concept so I can use it. The concept of the renal corpuscle. When people talk about a renal corpuscle, that means the glomerulus with its afferent arteriole and efferent arteriole and the Bowman's capsule. So all of this portion here, which I'm now surrounding with a box, and I'll actually even colour that in, highlight it in. So a nice box highlighted in around it now. That is what is meant by a renal corpuscle. So the juxtaglomerular apparatus is going to involve the renal corpuscle and the initial portion of the distal convoluted tubule, which, as we're going to see, is actually very closely located with the renal corpuscle. Okay, so let me now draw this then. So I'm going to draw, starting off with the afferent arteriole, glomerulus, and efferent arteriole, larger this time, and then we're also going to look at the structure of Bowman's capsule in a bit more detail, and then we'll add on the proximal portion of the distal convoluted tubule. Okay, so a bigger picture then, firstly, of the afferent arteriole, the efferent arteriole, and the glomerulus in between. So here is the efferent arteriole, here is the afferent arteriole here. So we're going to have capillaries in the middle here, like so. And these are the actual glomerulus. Excellent. So what do I now want to show on this picture that I didn't show previously? Well, I want to show you the different layers of these blood vessels, particularly the afferent arteriole and the efferent arteriole. I'm not interested in the endothelium. Okay, so I'm not going to try and draw the endothelium on here. The endothelium will of course consist of a basement membrane with endothelial cells sitting on it. I'm not going to draw that because it will just make the picture more complicated than it needs to be, and also it will be actually quite difficult to draw endothelial cells on this picture because everything's so crowded. What I am drawing at the moment, you can imagine, if you like, represents the endothelium, or you can imagine that it is the basement membrane of the endothelium, and then you'd have endothelial cells sitting on here, and I'm actually going to break the rules that I just talked about. I'm going to draw a little endothelial cell on here. Okay, I think it'll just make it simpler actually. So here are some endothelial cells, and you can imagine continuing this on, putting endothelial cells all over where I've coloured in orange. So you'd have an endothelial cell over here, etc. You'd have endothelial cells over the entire basement membrane. Okay, so I'm not going to draw them all. Right, so we've got our endothelium here then, and I'll just put a few on here. Now, in the case of the capillaries, that's just going to be the entirety of it. Okay, capillaries are very simple blood vessels. They just literally consist of an endothelium, endothelial cells with a basement membrane underneath them, occasionally a few pericytes. The afferent and efferent arterioles, however, they're going to have some very special cells surrounding the endothelium. which are analogous to smooth muscle cells, vascular smooth muscle cells, but which have an incredible function in the renin-angiotensin-aldosterone system, a function that normal vascular smooth muscle cells around an arteriole, let's say in the skin, do not have. Okay, and these special variants of vascular smooth muscle cells, they are known as juxtaglomerular cells or granular cells. We will most likely refer to them as granular cells, but I might refer to them as juxtaglomerular cells. So around the endothelium then of the afferent arteriole, and to a lesser extent, but they're still there, around the efferent arteriole, you're going to find these granular cells, and let me draw a few of them bluntly on here. So we'll have them around this afferent arteriole like so, and I'll divide this up now into the cells. We'll have 4 on each side, and we'll have a few over here, so I'll just draw 2 over here like so. So I'll give them nuclei, so here are their nuclei, and these are the cells that are actually going to release the enzyme renin in response to mean arterial pressure going down. And our task now is to understand how these cells actually find out that mean arterial pressure has gone down. But first, let's just complete the juxtaglomerular apparatus, because these cells are not the entirety of the juxtaglomerular apparatus. They are one of the key components of it, okay, probably the key component if you like. They're the ones that actually produce the renin and release it then into the bloodstream, but they're not the only component of it. So the granular cells are our first example and most important part of the juxtaglomerular apparatus, and they are these adaptive vascular smooth muscle cells, and they do still have the ability to contract and control the diameter of the lumen of the afferent and efferent arteriole, but their ability to secrete renin is what we're really interested in here. Okay, so those are some cells that surround the endothelium of the afferent arteriole and the efferent arteriole, and they form part of the juxtaglomerular apparatus. Now, what do I want to do next? I'll add on Bowman's capsule next. So we're going to do Bowman's capsule now in more detail. So I've discussed that Bowman's capsule, it is a space known as Bowman's space with, Bowman's capsule is actually the tissue that surrounds the space. So Bowman's capsule is this tissue that surrounds Bowman's space, and it's going to have a parietal portion and a visceral portion. So this is an important concept. The parietal portion of Bowman's capsule is the portion here that doesn't actually surround the glomerulus. The portion that surrounds the glomerulus here is known as the visceral Bowman's capsule. So if I draw it on like so, this portion here is known as the parietal Bowman's capsule, and this portion here is known as the visceral Bowman's capsule. So this is the new concept that I want to show you, the difference between parietal Bowman's capsule and visceral Bowman's capsule. And this is very similar to the concept of the parietal pleura and the visceral pleura, or the parietal pericardium and the visceral pericardium, or if you want to take it more complicated, the parietal peritoneum and the visceral peritoneum, but the peritoneum's ridiculously complicated, so that's not a good example to use. The pleura is the nice, simple example. Okay, it's that same principle of you've got a sac, if you like, which is the pleural sac, and you've got fluid inside that, and then what you've done is you've forced the lung into that sac. You've pushed it in. The lung ends up coated in the visceral pleura, and then the pleura that's on the outside, and I need a picture to explain this. So if we're using the analogy of the lung, you can imagine creating this sac then, okay, like a balloon filled up with fluid, and the fluid here is going to be the pleural fluid. And then what you do is you take the lung, and I'll just draw my lung here, like so. You take your lung and you push the lung into the side of this sac here, and you end up with the lung getting coated by this layer of the side of the sac. So what ends up happening is something like this. So here's the sac, like so, with the lung now sitting here, and then what you do is you just sort of mold this sac around the lung, and then this portion of the sac wall that is actually surrounding the lung, that's known as the visceral pleura, and then the portion that isn't surrounding the lung, this portion here, is known as the parietal pleura, and then there's the pleural fluid, which was the stuff initially inside the sac, in between those two layers. So it's the same sort of concept here. You can imagine that Bowman's capsule is like a sac with Bowman's space in the middle, and then you sort of push the glomerulus into the side of it here to form a visceral layer that actually covers the glomerulus, and then the parietal layer here. Now, the parietal layer of Bowman's capsule, this consists of a squamous epithelial. It's a squamous epithelium. Okay, so it consists of simple squamous epithelial cells. So I'll just draw these like so. So here they are, these simple squamous epithelial cells here, like so. And I'm on some of them drawing a little sort of extension where the nucleus will be here, because we've managed to go through their nucleus. Some of them we won't see the nucleus because we've just taken a cross section. And then we'll have another one here. I'll put the nucleus there. Okay, so you've got these simple squamous epithelial cells, which will be sat on a basement membrane as always. All epithelial cells are always sat on a basement membrane to keep them where they are, so they don't just fall off. And that makes up the parietal edge of Bowman's capsule then, and surrounds the Bowman's space here. And we'll talk about what's over here now. So then what this will turn into is the epithelial lining of the proximal convoluted tubule here, and these cells are quite different from the cells of the parietal Bowman's capsule. They're going to be cuboidal epithelial cells. Again, they'll be sat on a basement membrane, but they're going to be cuboidal in nature, not squamous. So here are their nuclei, like so. And these are the renal tubule cells that line the proximal convoluted tubule, and they'll have the role of reabsorbing lots of the stuff that's good that is in the glomerular filtrate into the bloodstream, and secreting stuff that we want rid of from the bloodstream into the fluid that is passing through the lumen of the proximal convoluted tubule here. So I'll colour these in, not in blue. I don't want to use blue, sorry about the shape. What colour do I want to use? I'll use yellow. Don't want them to look too similar to the granular cells that are so important for the juxtaglomerular apparatus. The proximal convoluted tubule renal cells here, these are not important for the juxtaglomerular apparatus. I'm just doing this for background, to put it in its place here. So here are the proximal convoluted tubule cells. Then let's put on the visceral Bowman's capsule. Now, the visceral Bowman's capsule is made up of a bunch of bizarre looking cells known as podocytes. So this is a reasonably difficult cell to get your head around. So the visceral Bowman's capsule is made up of these cells known as podocytes. Now, podo refers to feet. Cyte means cell. So these are cells with feet. And podocytes actually look like this. They have a cell body with the nucleus in, and then they have these processes coming off them which then have foot plates, like so. So they'll have multiple of these. So here's another one, and then here is a third one here, and a fourth one here. So these cells have a cell body here, and then they have these processes coming off, and then they've got these supports, effectively, these feet, and these are known as pedicels. Okay, so podocytes have these pedicels coming off them. And what's actually going to happen is you're going to make the visceral Bowman's capsule out of the pedicels of podocytes. So it's going to be like this. You'll have a podocyte here, and then coming off the podocyte you'll then have pedicels, like so, which are going to make up the visceral Bowman's capsule. So there is one podocyte here, and its pedicels, as you can see, are making up the visceral Bowman's capsule. And the pedicels are going to be attached to the basement membrane of the endothelial cells of the capillaries of the glomerulus here. So if I want to draw another one, here are some more pedicels here, and then we'll have the podocyte that is not actually itself part of the visceral Bowman's capsule. You can see that it's sitting in the Bowman's space actually. It's the pedicels that are forming the layer, okay, of the visceral Bowman's capsule. So there's another podocyte, and I'll just draw one more for completion here. So some more pedicels here, and the glomerular filtrate is mainly going to pass through the gaps between the pedicels, which don't form an absolutely tight barrier. Okay, so whoops, I'm just re-straightening this up. So here is my final renal podocyte that I'm going to draw, like so. Okay, right, so that's what the visceral Bowman's capsule then is made up of. And the podocytes, they're not part of the juxtaglomerular apparatus either. I'm just doing this for some interesting background to put it all in context. So there is Bowman's capsule done in a little bit more detail. Now what I want to show you is the other key component of the juxtaglomerular apparatus, which is the cells, certain important cells in the first portion of the proximal convoluted tubule, and the distal convoluted tubule. And the distal convoluted tubule, as I said earlier, it actually ends up very close to the renal corpuscle here. So here is now what I'm going to claim is the first portion of the distal convoluted tubule, located nicely close to the renal corpuscle. And the apparatus here, the cells of special cells of the early portion of the distal convoluted tubule near these granular cells here, this is what is meant by the juxtaglomerular apparatus. So this is a cross section of the distal convoluted tubule. So even though on this picture, for simplicity, I showed the distal convoluted tubule far, far away from the renal corpuscle. Imagine looping this round, you can imagine how to do it, I hope. Twist it round, bring it back over here so that it's close by the renal corpuscle. It's not unimaginable how this can actually arrange itself in three-dimensional space. So here then is the distal convoluted tubule, and in the early portion of the distal convoluted tubule, some of the renal tubule cells there, the epithelial cells that line that tube, have a very special function, and these cells are known as the macular denser cells. Now, not all of the cells of the proximal portion of the distal convoluted tubule are macular denser cells, but some of them are, and these cells are the ones that are going to be part of the juxtaglomerular apparatus. So these cells that I'm drawing on here now, these are supposed to be the macular denser cells. Okay, and why is it called macular denser? Well, macular means spot, denser means dark. So this is a portion that histologically shows up dark of the early portion of the distal convoluted tubule. So that's the way that you can actually identify these special cells of the juxtaglomerular apparatus that are in the early portion of the distal convoluted tubule here. So I will colour them in red here, but know that their name just refers to how they can be histologically identified. Okay, so this then is the juxtaglomerular apparatus, these macular denser cells along with these granular cells. Now let's actually talk about its involvement in the Renin-Angiotensin-Aldosterone system. A lot of time spent on the structure of this. Let's actually talk about what this is going to do. So overall, the Renin-Angiotensin-Aldosterone system starts when the granular cells release renin into the blood. And as I said earlier, there is a basal amount of activity of the Renin-Angiotensin-Aldosterone system, and this is because these granular cells, they do release a bit of renin all the time, and it's that basal activity that's going to be inhibited when mean arterial pressure goes too high. We are initially discussing what happens when mean arterial pressure goes too low because it's much more dramatic, much easier to understand. So, when mean arterial pressure goes far too low, these granular cells are going to start releasing much, much more renin into the bloodstream. Okay, so renin, I will come to this later on, it's going to be an enzyme that is going to then activate the next component of the Renin-Angiotensin-Aldosterone system. So I'll draw it in the way enzymes are classically shown with the active site shown here. Okay, so renin is an enzyme that's going to be released by these granular cells into the bloodstream. Okay, right. Now, what we want to understand then is how does mean arterial pressure going down activate these granular cells to release more renin into the bloodstream? What is the actual pathway for activating the granular cells to release renin into the bloodstream? Well, it involves the macular denser cells, but it doesn't have to involve the macular denser cells. There are actually 3 ways in which when mean arterial pressure goes down, you can activate these granular cells to release renin into the bloodstream, and I want to now go over these 3 ways. So let's say that mean arterial pressure has gone down. How are we going to activate increased release of renin by these granular cells into the bloodstream to increase our activation of the Renin-Angiotensin-Aldosterone system? Well, one way is that actually sympathetic nervous system output to the granular cells goes up. So I haven't mentioned this so far. These granular cells, they are innervated by postganglionic sympathetic nervous system neurons. So let's say this is the axon terminal of one of these postganglionic sympathetic nervous system neurons. And actually, a part of the baroreceptor response that I didn't talk about is that when mean arterial pressure goes down, you're also going to increase sympathetic output to the granular cells of the juxtaglomerular apparatus. So we talked about in the baroreceptor response, when blood pressure goes down, it results in increased sympathetic output from the cardio-acceleratory center and the vasomotor center, and that's going to increase cardiac output and increase total peripheral resistance. But actually you get other portions of the sympathetic nervous system activated as well. Their output is increased. And this is an interesting example of another one. You're going to increase sympathetic output to these granular cells, and indeed when mean arterial pressure goes too high, we know that then the sympathetic nervous system is toned down to the heart and to the vasculature, and it's also going to be toned down to these granular cells, which is going to be one of the ways that we reduce the activation of the Renin-Angiotensin-Aldosterone system when mean arterial pressure is too high. So, going back to mean arterial pressure being too low then, we have sympathetic output to these granular cells. So this postganglionic sympathetic nervous system neuron then, it's going to release noradrenaline as usual onto the granular cells, and just drawing a big picture of our granular cell here. So here it is. The granular cells, they actually have the beta-1 adrenoreceptor. They're one of the other examples of tissues in the body where the beta-1 adrenoreceptor is actually used. Beta-1 adrenoreceptors are on cardiomyocytes, they're also on granular cells, but you struggle to find other examples of tissues that actually contain this adrenoreceptor on the surface. So it's one of the other notable examples. Everyone knows about the heart, but here's another example of where the beta-1 adrenoreceptors are actually made use of. Okay, so noradrenaline will bind to the beta-1 adrenoreceptor, and it triggers the release of renin into the bloodstream. So it'll activate renin release into the bloodstream by our granular cell here. So I'll just give it a nucleus. Okay, so that's one of the ways then that mean arterial pressure going too low can increase renin release by the granular cells. It's going to increase the sympathetic output to the granular cells, and that will activate increased renin release. And of course, we can already think in our heads that mean arterial pressure going too high will reduce sympathetic output and thereby reduce renin release, but we'll come on to that later on. Okay, so that's one of the ways then that too low blood pressure can activate renin release. In addition, the granular cells are actually baroreceptors themselves, so they are sensitive to the blood pressure in the afferent arteriole and also in the efferent arteriole, so they are baroreceptors themselves. Now, clearly there is a pressure in these arterioles. There's a force with which the blood is pushing against the wall of the arteriole. The higher the arterial blood pressure is, the more that pressure in the afferent arteriole and the efferent arteriole will be. So they do actually have a means to get information about mean arterial pressure. So if the pressure in the afferent arteriole and the efferent arteriole falls, that indicates that mean arterial pressure might have fallen. So these are baroreceptors themselves, so if they notice that the pressure in the arteriole has decreased, they will therefore increase their renin release. So if pressure in the arterioles has decreased, so pressure decreased, then this is going to lead to renin release going up. So there's another way in which you can increase renin release when mean arterial pressure falls. And again, you can imagine that if mean arterial pressure was to go too high, that would mean that the pressure inside the arterioles was increased, and therefore these granular cells would decrease the amount of renin that they were going to be releasing into the bloodstream. Finally, let's now talk about the macula densa and its involvement in coordinating renin release or controlling renin release by these granular cells. So this is the more complicated one. The macula densa cells, they have an ability to measure the glomerular filtration rate, the GFR. So this is a very important concept in physiology. GFR means glomerular filtration rate, and this refers to how much glomerular filtrate do you actually produce in a minute. And usually in a healthy person, the glomerular filtration rate is usually about 125 milliliters per minute. Okay, now what this means is that usually you are producing 125 milliliters of glomerular filtrate every minute. Now, in each nephron, you're going to be producing a tiny little amount of glomerular filtrate every minute. Now, if this was to decrease, so if glomerular filtration rate was to decrease, then the macular denser cells are going to be able to detect that, and there is a very clever way in which this occurs. So if you imagine now that the amount of glomerular filtrate that is being produced in a minute by a single nephron now, this of course means how much is produced by all of the nephrons, but now let's talk by a single nephron. So if we're talking about a single nephron here, it's going to produce a certain amount of glomerular filtrate every minute. Now if you imagine that that was to decrease, then the rate at which fluid is flowing through the nephron would decrease. And the macular denser cells are actually going to have a clever way of detecting that. Now, why is that going to have bearing on mean arterial pressure? Well, if mean arterial pressure was to decrease, we've talked about how that means that pressure inside the arteriole is going to decrease. And what's also going to decrease is the amount of blood that is actually flowing through here. But because, of course, mean arterial pressure has gone down, and that's the driving force for blood to flow through any blood vessel. So, both the fact that the pressure in here is decreased and the fact that the amount of blood that's actually flowing through here in a minute has decreased is going to lead to glomerular filtration rate going down. So overall, if mean arterial pressure goes down below what it should be, glomerular filtration rate is going to go down. The amount of glomerular filtrate that is being produced in a single nephron in a minute is going to go down. And that means that the rate at which fluid is flowing through the nephron is going to go down. And that, believe it or not, is detectable by the macula densa. So the cells of the macula densa, they are capable of sensing the sodium chloride level in the fluid that is arriving at them. So they can detect what is the sodium chloride level in this fluid that is arriving. Now, sodium chloride is a very, very important solute inside the extracellular fluid of the body. It's one of the major dissolved solutes in the extracellular fluid of the body. So, there is going to be a lot of sodium chloride in the glomerular filtrate that we initially produce, and that's going to need to majorly be reabsorbed by the kidney. Now, by the time the glomerular filtrate actually gets to the distal convoluted tubule, most of the sodium chloride has already been reabsorbed. In fact, only 8% of the sodium chloride that was initially in the glomerular filtrate here is still present when it arrives at the early portion of the distal convoluted tubule where the macula densa is. So the macula densa will be used to seeing that concentration of sodium chloride. If you now imagine that fluid is flowing through the kidney more slowly because glomerular filtration rate has gone down, because mean arterial pressure has gone down, and therefore the pressure in these glomerular capillaries has gone down, which is the driving force for fluid to come into the Bowman's capsule, and also the flow through the glomerulus has gone down, so that will also reduce the amount of fluid that is moving into the Bowman's capsule. So if you can imagine now that fluid is moving slower through the nephron, then the early portions of the nephron here, the portions prior to the early portion of the distal convoluted tubule, are going to get a greater opportunity to remove sodium chloride. So, you know, if the fluid's moving more slowly, you can imagine that it's going to remove more sodium chloride from there. If you imagine these cells are grabbing sodium and chloride ions from the fluid that is passing through here, if the fluid's passing very slowly, they'll be able to grab a lot more of them out of this fluid than if it's passing at the more normal rate. So this means then that the amount of sodium and chloride ions that actually end up in the fluid arriving at the distal convoluted tubule is below what it would normally be. So the macular denser cells can therefore interpret what the flow rate of the fluid through the nephron is by the amount of sodium chloride that is meeting them, and therefore they can get this insight into what glomerular filtration rate is doing, and glomerular filtration rate is their insight into what mean arterial pressure is doing. So the macular denser cells, they're really measuring glomerular filtration rate via sensing sodium chloride level, but glomerular filtration rate depends on mean arterial pressure. So if glomerular filtration rate goes down, and therefore the amount of sodium chloride in the lumen of the early distal convoluted tubule where the macular denser cells are goes down, the macular denser cells are going to send a signal to the granular cells telling them to release renin and start the renin-angiotensin-aldosterone system. Now, what is this signal? What is the nature of this signal? Well, there's two components to this signal. There's one component which is that they're going to release a specific molecule when the sodium chloride level that is reaching them actually goes down. And the second component is that they're going to stop sending a molecule that they usually send when sodium chloride level is normal. So let me firstly talk about the molecule that they usually send. So the macular denser cells are normally releasing adenosine molecules, which is just an ATP molecule with the free phosphate groups removed. It's the nucleoside, which is a ribose sugar bound to an adenine molecule. So the two of the portions of a RNA nucleotide, but without the phosphate group. So they're going to release adenosine molecules, and these adenosine molecules are normally the message to the granular cells to keep calm and do nothing. I always actually think when thinking about this, I always think of Voldemort in the 4th Harry Potter book, screeching, "Do nothing unless I command," in the voice of Stephen Fry. Just a glimpse into my childhood there. Okay, so that's effectively what the macular denser cells are doing with this adenosine, however. It's a cry of, "Do nothing, everything's fine." So the macular denser cells, they usually release adenosine, and the adenosine goes over to the granular cells and inhibits the release of renin. So adenosine inhibits the release of renin. So this is the normal molecule that the macular denser cells are secreting, and it's a cry of, "Do nothing, sodium chloride level here is fine, glomerular filtration rate therefore is fine." This command is going to go suddenly if sodium chloride level reaching here goes down, and therefore glomerular filtration rate has gone down. So the macular denser cells will stop releasing adenosine, and this will therefore no longer be giving this cry of "do nothing" to the granular cells. In addition, they also send a command of "do something," and that command is believed to be in the form of prostaglandin E2, although there is still debate about specifically which prostaglandin is used by the macular denser cells, but most people, I think, believe that prostaglandin E2 certainly is very important in this command signal. So, the macular denser cells will start releasing prostaglandin E2 if they have sensed a low level of sodium chloride reaching them, which therefore indicates that glomerular filtration rate is too low and mean arterial pressure is too low. And prostaglandin E2 will activate the release of renin. Okay, so this now causes the release of renin. So let me summarize that, because it's a complicated concept. So we've talked about the fact that mean arterial pressure determines glomerular filtration rate. Glomerular filtration rate, the number, means how much glomerular filtrate all the all the nephrons of the kidneys are actually producing in a minute. We're thinking in terms of a single nephron, so a single nephron will be producing a certain amount of glomerular filtrate in a minute. If that was to go down, then the flow through the kidney would go down, and therefore the amount of sodium chloride that's actually going to reach the cells of the early portion of the distal convoluted tubule is going to go down, because the cells of the prior portion of the nephron to the early distal convoluted tubule are going to have more of an opportunity to remove sodium chloride molecules from the filtrate as it passes more slowly by them. Okay, that is how the macular denser cells determine what glomerular filtration rate is doing in that nephron. And if they find that it's going down, then they're going to send the command to the granular cells to release renin. And there's two components to how this command works. Usually they send a command of do nothing, sodium chloride level reaching us is fine, which is the adenosine molecules that they release. They're going to stop sending the adenosine molecules, so they stop their brake on the granular cells. They're also going to send a command to actually do something, which is prostaglandin E2, so they're going to start releasing prostaglandin E2, which they don't usually release, which will activate the granular cells to release renin. So all of these things then are going to start occurring when mean arterial pressure goes too low. You're going to get the baroreceptor response, increasing sympathetic output to the granular cells to increase their release of renin. The granular cells themselves are baroreceptive to the pressure in the arterioles, and they're going to therefore note that there's something wrong with mean arterial pressure and we should release more renin. In addition, the macular denser cells will tell them that there's something wrong with glomerular filtration rate, it's too low, therefore mean arterial pressure is too low, and that will activate the release of renin. Now let's talk about what would happen if mean arterial pressure was too high, the opposite problem. Well, if mean arterial pressure was too high, the baroreceptor response we know will decrease sympathetic output to the heart, decrease sympathetic output to the vasculature, and it's also now going to decrease sympathetic output to these granular cells, so in our wider view of how this baroreceptor response is actually occurring. So that's one of the things that's going to cause them to release less renin. In addition, the granular cells will be receptive to the pressure themselves, and pressure of course in the arteriole will now be higher than before, and that's going to inhibit their release of renin. So pressure will be too high, and that will also cause them to want to release less renin. Finally, of course, if mean arterial pressure is too high, then glomerular filtration rate is going to go up, so the macular denser cells are now going to be receiving too high a concentration of sodium chloride in the fluid that's reaching them. Okay, because if the fluid's moving quicker through the nephron, there's less opportunities to reabsorb sodium and chloride ions. So now what's going to happen is they're certainly not going to be releasing prostaglandin E2, they're going to start releasing more adenosine, more command of do nothing in Voldemort's voice, and that will be inhibiting the granular cells further and preventing their release of renin. So renin release goes up from the basal tone level if mean arterial pressure is too low, and it goes down if mean arterial pressure is too high. So we increase the activation of the Renin-Angiotensin-Aldosterone system if mean arterial pressure is too low, and decrease activation of the Renin-Angiotensin-Aldosterone system if mean arterial pressure is too high. We will have a break here. In the next video what we'll do is continue the story on, and the story gets much more simple from now on. It's just one thing causes another thing, causes another thing, causes another thing. It becomes a beautiful linear flow chart, but we'll do that in the next video.